Hi, my name is Cynthia Owsley. I'm doing a presentation on vision or visual function for the Ryan Initiative for Macular Research. I'll be doing part 1, focused on certain visual functions, and Max Val will be doing part 2 on other visual functions. To get a comprehensive picture, please listen to both. Probably the most meaningful issue to patients who have AMD is the quality of their vision, both what it is currently and the prognosis for the future. Visual function as endpoints or outcomes are critical in clinical trials, but also in longitudinal observational studies. There are many candidate visual functions to select from when designing a study. Psychophysical methods or behavioral methods are used to measure vision. As you will see in reading the research literature, some visual functions have been commonly used in studies, while others are more infrequent. When selecting a visual function as an endpoint or outcome, it is important for the psychometric properties to be strong. What am I referring to here? These properties are repeatability, validity, and responsiveness to intervention. I won't be presenting these detailed psychometric features for each of the visual functions I'm going to discuss today because of the time. However, when you are designing a study, please research and document the presence of these features in the visual function you are selecting. In addition, it's also important for visual functions to be as brief as possible in order to fit well into a clinical trial protocol. It is also important to keep in mind that strong candidates for studying or preventing end-stage AMD will most likely be different than those strong candidates for studying early disease or for preventing AMD. In this presentation, I'll be discussing the following visual functions: visual acuity, low luminance visual acuity, or low luminance deficit, spatial contrast sensitivity, rod-mediated dark adaptation, reading, and mobility. Max will be discussing light and dark adapted perimetry and microperimetry under steady-state conditions and tests of color vision. Christine Curcio's work indicates that it is important to remember that rods dominate the macula. The fovea, of course, has a very high density of cones, but the predominant photoreceptor in the macula are rods. Also from Christine's work in the 1990s, we know that rod loss, not cone loss, is characteristic of aging. During aging, rods decrease by 30% near the fovea, but cones are protected. During the early progression of AMD, we know from Christine's work that rods are vulnerable in early and intermediate AMD, but cones are not vulnerable. In end-stage AMD, the cones degenerate. The last surviving photoreceptors, though, in end-stage AMD in the macula are largely cones. I'd like to begin my discussion of visual function outcomes with visual acuity. Acuity is measured under best correction conditions, meaning after refraction. The optotype is letters. The ETDRS chart, pictured here on the right, was the first well-designed acuity chart for use in research. It was based on principles identified by the National Academy's Committee on Vision, and then further developed by Bailey and Lovey in Australia, and then further implemented by the National Eye Institute for use in studies on diabetic retinopathy. ETDRS acuity, as it is called, is measured via a light box to standardize the illumination for a photopic or daytime light level. The chart uses an invariant number of letters per line, that is 5 letters per line. The scale is logarithmic at 0.1 logmar per line. With the patient 4 meters away from the chart, a coordinator can test patients with visual acuity from 2200 to 2010. A nice feature is that for patients with very poor acuity, say worse than 2200, you can move the patient closer and still get a measure. The ETDRS charts are widely commercially available and are commonly used in clinical trials. In the late 1990s, Roy Beck of JUB in Tampa, Florida, developed an electronic computerized method for measuring visual acuity. The test is repeatable and, importantly, was validated against the ETDRS chart. An important feature is that single letters are presented. This prevents the patient from getting confused when reading across a line. For example, it prevents the patient from accidentally jumping lines. The EVA was initially available from JUB, but it is now commercially available. It is also commonly used in studies. Visual acuity is highly useful for studies on advanced AMD, since in the course of AMD, advanced AMD is when visual acuity actually declines. On the top, we see that it was very useful in the CAT study for demonstrating the equivalence between these two treatments for neovascular AMD. It is also important to point out that visual acuity is not really useful for studying early and intermediate AMD. On the bottom, we see data from All-Star 2 for patients with normal aging and early and intermediate AMD. As you can see, visual acuity was approximately 20/20 in all groups, with the standard deviations also being very similar. The Morse Field Acuity Test, or MAT, was developed in the UK based on principles identified by Howland. It is also known as the vanishing optotype test. The letters have a pseudo-high-pass design. The vanishing optotype nickname is derived from the fact that detection and resolution thresholds are very similar in the fovea. What this means is that the letter vanishes almost as soon as the resolution threshold is reached. This chart is more commonly used in the UK and in Europe. For example, this chart is being used in the MacuStar study. The developers of the chart presented some evidence that this test is more vulnerable to cone loss in AMD, and thus could make it a useful candidate for studying advanced AMD. Low luminance visual acuity is measuring acuity under mesopic conditions. Patients view an acuity chart through a 2.0 neutral density filter. In some studies, the neutral density filter is higher than 2.0, but the idea, or higher or lower than 2.0, but the idea is that the chart is viewed at a mesopic light level. The difference in visual acuity at a photopic light level and measuring it under mesopic conditions is called the low luminance deficit, or LLD. Janet Sunnis developed this acuity test back in the late 1990s and revealed visual deficits in geographic atrophy that were not revealed with a photopic acuity test. Rosenfeld's group, shown there on the right, has recently used the test to demonstrate that low luminance visual acuity and the low luminance deficit are associated with decreased central macular choriocapillaris perfusion. Low luminance acuity and low luminance deficit can be useful in studying advanced AMD, such as geographic atrophy. The original tests of the contrast sensitivity function, also called the CSF, were measured as a function of spatial frequency. One of the first demonstrations of impaired contrast sensitivity is a study by Sjöström and Friesen in Sweden. This study was performed in AMD patients. At the time, AMD was still referred to as senile macular degeneration, which is pictured there on the left if you can read the caption. The challenge with measuring the whole CSF is that it is very time-consuming, and brief vision tests are typically preferred in clinical trials. Therefore, Pelley and Robson developed the letter contrast sensitivity chart as pictured at the right. This letter chart does not measure the whole CSF. Rather, it provides an estimate of sensitivity near or just below the peak of the function. Pelley-Robson charts have been commonly used in studies. More recently, Arditi developed the Mars contrast sensitivity chart. It was designed to overcome some of the practical limitations of the Pelley-Robson chart. The Pelley-Robson chart can yellow over time and hangs on the wall. The Mars chart is handheld by the patient, is plastic, a hard plastic, and it's easy to clean. It also does not yellow over time. As you can see, it looks very similar to the Pelley-Robson chart. It's important to point out that the Mars chart has been validated against the Pelley-Robson chart, meaning it provides highly similar data. It is commercially available and is also increasingly used in studies because of its practical features. As indicated by the All-Star 2 study, contrast sensitivity using the Mars chart remains relatively stable into early AMD, with a modest decline in intermediate AMD. It is also worth pointing out that when Mars contrast sensitivity is measured under mesopic conditions, sensitivity impairment is accentuated, which you can see at the bottom of the table. Another recent development is the quantitative CSF, abbreviated QCSF. It is also sometimes called the quick CSF. An example from the quick CSF is pictured on the top. It was developed by Lesmus and colleagues. It uses a Bayesian adaptive procedure to increasing the efficiency of measuring sensitivity over a broad range of spatial frequencies. Rather than using sine wave gratings or Gaussians, the QCSF uses letters. Scholl and colleagues, pictured there on the bottom, have recently shown that in intermediate AMD, the most noteworthy impairment is around the peak of the CSF or lower. The literature on the QCSF has been growing in recent years. An issue that hopefully will be resolved soon is whether the QCSF in early and intermediate AMD provides information beyond that provided by the Mars chart or the Pelley-Robson chart. These latter two charts provide information around or just below the peak of the function in a very brief fashion. Rod-mediated dark adaptation refers to the recovery of light sensitivity by rods after a photobleach. This can be either a prolonged bleach over time or a flash bleach. I showed in 2001 that delayed dark adaptation is characteristic of AMD even in its early stages, as shown on the left. The first All-Star study showed that delayed dark adaptation in normal older adults doubles their risk for early AMD 3 years later. Kukris's work, displayed on the right, showed that dark adaptation delays become longer with increasing disease severity. Delayed dark adaptation can be measured with a measure called the rod intercept time, which is the duration of time to recover to the second component of rod-mediated dark adaptation. The bottom table shows in the All-Star study how rod intercept time gets longer as you increase from aged normal to intermediate AMD. Rod intercept time averaged 12 minutes in aged normals and averaged a much higher number, 29 minutes, in intermediate AMD. Reticular pseudodrusen recently have been referred to as subretinal drusenoid deposits. Both terms are used. RPD or SDD exacerbate dark adaptation delays. This is shown here by Kukris's group, and also below that by our All-Star 2 study. It is important to point out that even when SDD or RPD are absent, drusen are also associated with dark adaptation delays. The delays due to drusen alone are typically smaller than for eyes with SDD. A question of interest is where are dark adaptation delays on the retina in AMD most pronounced. As it turns out, dark adaptation delays are the most pronounced the closer you get to the fovea, as shown here by Geimer's group on the right and also in our All-Star 2 study. Our recent work in All-Star 2 has shown that dark adaptation as a functional endpoint is actually correlated with structural biomarkers. For example, slowing in dark adaptation in AMD has been associated with the IZ and IZ thickness, SDD area, choriocapillaris flow deficits, and fluorescence lifetime imaging ophthalmoscopy, or FLIO. This is a reminder to those designing clinical trials that it's best to select functional endpoints that are correlated with structural biomarkers. This will enhance the likelihood in a trial that you will identify both a significant structural outcome and a significant functional outcome. I'd like to tell you about a couple of outcomes that consist of performance tasks of daily living. The first is reading. MN-Read is a test developed by Gordon Legg's group at the University of Minnesota. Its repeatability has been widely studied, as illustrated here. It consists of single sentences of text, not longer paragraphs. It can be administered on an iPad and is commercially available. Actually, 4 measures are provided by MN-Read. These are reading acuity, maximum reading speed, critical print size, and reading accessibility. It has been formally translated into a number of languages as mentioned on this slide. You could read more about it at the web link. Since reading is driven by both acuity and contrast, the MN-Read test could be useful for studying late intermediate AMD as it transitions to advanced AMD. IREST refers to the International Reading Speed Text and was developed by a multi-site effort in several countries. It is available in 17 languages, which is highly suitable for multi-center studies done at various locations around the world. Unlike the MN-Read, it does not use sentences, but rather paragraphs. The outcome measure is reading speed expressed as words per minute. MacuStar uses iRest. They have found that it does not discriminate among normal controls in early and intermediate AMD, but it does discriminate between intermediate AMD and advanced AMD. This underscores that reading tests such as iRest and MNRead can be appropriate as outcomes later in AMD. I'd like to now tell you about another daily life test that could potentially serve as an outcome measure in AMD studies, namely mobility. The test pictured here is called the Multi-Luminance Mobility Test. It was actually developed for use in the Phase 3 RPE65 gene therapy trials. It was successfully used there as a beneficial outcome. It evaluates mobility performance in a real-life mobility course, or maze. Patients enter the maze, and they go through it from start to finish. They followed the arrows, as indicated in the diagram, and avoided obstacles in the path or obstacles that were adjacent to the path. An interesting feature of this test is that it could be performed at multiple luminance levels. Accuracy and speed were the outcome measures. Now, this test has not been used in AMD to my knowledge, but since AMD impacts rod photoreceptors in early and intermediate disease, and this test does manipulation of luminance level, it is interesting to consider it for possible future use in AMD studies. Another mobility test is called the MOST, or Mobility Standardized Test. This was developed by José Sahel's group in Paris. It was originally designed for use in RP trials as an outcome. Like the previous mobility test, it has good psychometric properties. However, the MOST is given through a virtual reality headset and is not a test that's done in the real world. However, in developing the test, the virtual reality device has been validated against the real-world mobility task. It can also be performed at several luminance levels. The outcome performance score is a combination of trial duration and mobility errors. Like the multi-luminance mobility test, there have been no published AMD studies using the MOST. However, several groups are planning to use it in observational AMD studies. Now I'd like to summarize the Part 1 vision presentation. First, visual acuity, low luminance VA, low luminance deficit can be useful in trials evaluating intermediate to advanced AMD. Recently, there has been increasing work on the quantitative or quick CSF in intermediate AMD. It would be useful to compare it to the performance of the letter contrast sensitivity charts as well. Rod-mediated dark adaptation will be useful for studying early to intermediate AMD and also for studying AMD prevention. This will be the case if pharma develops interventions targeted at these early phases of disease. Reading tests can be useful in studying intermediate AMD to advanced AMD. Mobility tests are exciting options and could be useful for studying mobility under lower luminance in AMD. While it has not been demonstrated, they could be useful in AMD studies. An important point is that structural biomarkers that are functionally validated will be most useful in trials because they have the potential of providing a coordinated functional and structural outcome. Here's my email address in case anyone has questions. Thank you very much for your attention.